The recent serological dissection of voltage-gated potassium channel complex antibodies between those which target LGI1 and are pathogenic, those which target the extracellular domain of CASPR2 and are also pathogenic, and those which target the intracellular aspect of some potassium channels merits investigations in terms of the potentially divergent immunological pathways. Knowledge of the high rate of adverse drug reactions to antiepileptics in patients with LGI1 antibodies led us to hypothesize that patients with LGI1 antibodies may well have an overexpressed HLA association, given we know that AED rashes also correlate with HLA associations. We were keen to search for HLA associations which could confer understanding of the etiology and also the immunological mechanism in these patients, particularly to understand the way antigens are presented by class 1 or class 2 molecules.
We aim to recruit a strictly phenotyped cohort of 111 individuals with antibodies to LGI1, CASPR2 or intracellular aspects of the voltage-gated potassium channel, the largest to date genotype-phenotype study of these rare patients. Patients underwent detailed clinical phenotyping via patient and relative interviews and case note review.
Patients with CASPR2 antibodies often present with a limbic encephalitis, which can be difficult to clinically distinguish from that occurring in patients with antibodies to LGI1. With an incidence of 0.3 million per year in the UK, CASPR2 antibody disease is rare. But we were able to recruit 31 patients to form a strong basis for this comparative study. We also included 3 patients with both antibodies. It's worth noting that the co-occurrence of these antibodies is more common than might be expected by chance. We also recruited 9 patients with antibodies to intracellular aspects of the VGKC only.
We used the Illumina Infinium platform to genotype the patient samples and then SNP to HLA methodology to impute HLA alleles at 8 classical loci. In addition, all DRB1, DRB4 and DQ alleles underwent intermediate resolution HLA typing using PCR sequence-specific primers. HLA alleles of 5,553 individuals forming the Oxford Biobank have been imputed using the same platform and were available for comparison as healthy controls.
So the clinical features first. The patient onset ages were around 60 years and most were male, with main presentations being encephalitis, epilepsy, a few with neuromyotonia and a few with irrelevant clinical syndromes. There was a high rate of coexisting autoimmune conditions, around a quarter in both the LGI1 and the CASPR2 patients. And distinctively, the LGI1 cohort had an elevated rate of drug reactions, particularly to antiepileptics, notably carbamazepine and phenytoin, and also to some antibiotics.
Therefore, we proceeded to analyse HLA associations. Consistent with previous smaller reports, almost all, 91% of LGI1 antibody-positive patients carried HLA-DRB171, and 13% were homozygous for this allele, compared to 2% of healthy controls. Other DQ class 2 alleles known to be in linkage disequilibrium with DRB171 were also overrepresented. Additional associations were found with 2 class 1 HLA alleles, and there were no significant HLA allele associations with subphenotypes.
CASPR2 HLA findings were in striking contrast to LGI1. A single risk allele was identified, HLA-DRB11-1, found in 48% of CASPR2 patients. Interestingly, none of the CASPR2 patients with the classically non-immune presentation carried this risk allele, giving it a 56% prevalence in the remainder of patients. Despite being only a very small group, of the 3 patients with both antibodies, all carried HLA-B44-2, C51, DQA31 and DQB31. Very intriguing finding. There were no significant HLA links for the VGKC only patients.
HLA haplotypes are important to understand in vivo relevance of HLA combinations, which may present LGI1 and CASPR2 antigens. The most frequent haplotype found in LGI1 antibody patients was HLA-DRB1-0701, DQA1-0201, DQB1-0202, but there was one class 1 haplotype associated with LGI1 antibodies, HLA-C0602, B5701. Again, there was a strong contrast between LGI1 and CASPR2, in which there was only one associated HLA class 2 haplotype, DRB1-1101, DQA1-0501, DQB1-0301.
This is the first comparative HLA analysis of LGI1 and CASPR2 autoantibody disease and shows marked and strikingly different HLA associations for these patients, suggesting that HLA status strongly predisposes to development of either LGI1 or CASPR2 antibody disease. However, the 4% to 28% rate of these HLAs in the healthy population implicates additional genetic, environmental or stochastic influences in the process of disease manifestation.
Overall, our data shows striking and distinct phenotype and genotype associations in patients with LGI1 and CASPR2 antibodies, and this immunogenetic dissociation supports the biochemical distinction between these two syndromes.